Very good afternoon, students. Yesterday, we already lost class. We already discussing about that is the RBC cells. Today class, we are going to discussing about white blood cells. Today class, we are going to discussing about white blood cells. Today we are going to discussing about white blood cells. The white blood cells are also known as leukocytes. The white blood cells are also known as leukocytes. The normal count of the leukocytes. The normal count of the leukocytes. Normal count of the leukocyte. 5,000 to 8,000 millimeter cube. The normal count of the white blood cells present in the human beings, 5,000 to 8,000 per millimeter cube. For example, this leukocytes increase in our blood. The leukocyte count is increases. It is nothing but leukocytemia. Leukocytemia. Increasing white blood cells. Increasing white blood cells is indicate leukemia. It is indicated leukemia. The white blood cells decreases. Leukocytopenia. Leukocytopenia means decreasing of white blood cells. Decreasing of white blood cells. Increasing white blood cells is known as leukocytemia. Decreasing white blood cells is known as leukocytopenia. Leukocytopenia. The shape of the white blood cells. The shape of the white blood cells. Amoeboid shape. What is the shape of the white blood cells? Amoeboid shape. The shape of the white blood cells. Amoeboid shape. Okay. Coming to the lifespan of the white blood cells. What is the lifespan of the white blood cells? The lifespan, for example, the white blood cells are present in the blood. The white blood cells are present in the blood. It is 4 to 8 hours. The white blood cells present in the blood. It is 4 to 8 hours. Or it is present in the tissues area or present in the capillaries. The white blood cells are present means it is 1 week. 1 week. Indicate 1 week. The different leukocytes, the different white blood cells present in our blood. Different leukocytes. Different leukocytes. Different leukocytes present in our blood. Okay, what is the leukocytes? Neutrophils and neutrophils, basophils, eosinophils, lymphocytes, monocytes. Different white blood cells present in the blood. Neutrophils, basophils, eosinophils, lymphocytes, monocytes. In the case of lymphocytes, the lifespan of the lymphocytes, days, months. It is indicated. Lymphocytes, days, months, okay, years, and also the complete lifespan. They present in the throughout the once you are going to death. Okay, it is also after the death. When up to the before the death, end of the lifespan complete means the lymphocytes are present. The lymphocytes, there are the lifespan is more. The lifespan is years. The lifespan is years. The general lifespan of the lymphocytes 48 hours or 1 week. But the lymphocytes, it is indicated. These are also under the category of the white blood cells. What is the lifespan? It is the years. The lymphocytes are indicated in the years. Coming to the special character of the white blood cells. The special character of the white blood cells. The special character of the white blood cells. blood components, it is the white blood cell. The special character, the white blood cells is act as a diapedicis. What is the white blood cells? Diapedicis. What is the white blood cells are there participated into the diapedicis process. Diapedicis process means, for example, it is the blood capillary. It is the blood capillary. Okay, the surrounding of the blood capillary is consist of tissues. The surrounding of the blood capillary is consist of the indicate tissue. It is indicated tissue. It is indicated blood capillary. It is indicated blood capillary. Inside the blood capillary, the blood is present. The blood consists of blood components, RBC cells and white blood cells. The sum of the white blood cells means monocytes. Monocytes. These monocytes, they can easily squeeze, easily penetrate, enter into the tissues. The tissues consist of any pathogen. Whenever the monocytes enter into this pathogen, it is consist of pathogens. Pathogens means microorganisms. Microorganisms present in the tissues. These monocytes are penetrate into the tissues. They engulf the foreign material. They engulf the microorganism. They capture the microorganism. These monocytes, they can easily penetrate. They can easily pass into the inside the tissues. The reason is the pathogens mean microorganism, bacteria, any fungi present in the tissues. Which cells they will fight against to these pathogens? It cells monocytes. The monocytes act as a macrophages. Macro means large. Phase means phag. Phase means cells. They will engulf the foreign material. Which cells are participated? Monocytes. Only the speciality of the diaphysis process takes place in the white blood cell. The special white blood cells are monocytes. These monocytes only participated into they engulf the foreign material. Coming to the different types of leukocytes. Different types of leukocytes. The leukocytes are classified into the different types of leukocytes. The white blood cells are also known as the leukocytes. The white blood cells are classified into two types. White blood cells are classified into two types. First white blood cell is known as the granulocytes. Second one, agranulocytes. Agranulocytes. A means absent. The granules, the proteins absent in the cytoplasm is known as the agranulocytes. Granulocytes means granules, proteins present in the cytoplasm is known as granulocytes. The granulocytes are divided into three types. A granulocytes are divided into two types. Granulocytes. The first one, neutrophils. Next one, basophils. Next one, eosinophils. Neutrophils, basophils, eosinophils. Agranulocytes. Lymphocytes. Next one, monocytes. Next one indicated monocytes. Coming to the first one, neutrophils. Next one, basophils, eosinophils. Next one, lymphocytes. Next one indicated monocytes. If you observe the neutrophils, the cytoplasma consists, the central body consists of nucleus. The nucleus is multi-lobed nucleus. How many lobes? Multi-lobed nucleus. Multi-lobed nucleus. Granules are present. The cytoplasm consists of granules. Granules is indicate nucleus. See here, nucleus. How many nucleus? It is consists of multi-nucleus. Consists of multi-lobed nucleus. Multi-lobed nucleus. Basophils is a spindle-shaped nucleus. What is a nucleus? Spindle-shaped nucleus. Eosinophils, bilobed nucleus. Cytoplasm is present. Bilobed nucleus. Lymphocytes, round-shaped nucleus. Monocytes this indicate horseshoe-shaped nucleus. Horseshoe-shaped nucleus or kidney-shaped nucleus. But in cytoplasma, no granules. That's what they indicate agranules. The cytoplasma, the granules or proteins are absent in the cytoplasm. Is known as agranulocytes. But here, granulocytes means the granules are present. Coming to the neutrophils. The neutrophils are the primary phagocytic cells. Neutrophils is act as a primary phagocytic cells. Primary phagocytic. Phagocytic means they engulf the foreign material. Coming to the next one. And monocytes. It is the efficient phagocytic cells. Secondary phagocytic cell. Phagocytic cells. They engulf the foreign material. Primary phagocytic cell, secondary phagocytic cell. That is the monocytes. Basophils, eosinophils, lymphocytes. These three are the non-phagocytic cells. Basophils, lymphocytes, monocytes. These three are indicated the non-phagocytic cells. Neutrophils, phagocytic cells. They engulf the foreign material. Okay, if you observe the neutrophils or basophils or eosinophils, how can you identify the nucleus of this neutrophils, basophils and eosinophils? You can add any stain, you can add any color, you can add any dye, you can observe these nucleus conditions. Okay, in these neutrophils, you can add either the acidic dye or basic dye. You can add acidic dye or basic dyes. Dyes means color, you can identify. For example, basophils, the basic dye you can add. The basic color you can add. What is the basic dye? Methylene blue you can identify. This one. Eosinophils you can add. That is the acidic dye. You can observe this eosinophils. Acidic dye you are added. Eosin dye. You can add eosin. You can observe eosinophils. You can add methylene blue. It is you can observe basophils. For example, you can add acidic acid or basic. You can add anyone. You can observe that is a neutrophils dye. Neutrophils dye. We are not added both dyes. Okay, coming to the lymphocytes. The lymphocytes, they will produce antibodies. The antibodies fight against to the microorganisms. The lymphocytes are participating in the immunity. The lymphocytes, what is the function of the lymphocytes? Immunity. They fight, they produce antibodies. They produce these antibodies only. They will engulf the foreign materials. Okay, angle of the foreign materials. Coming to the next one, basophils. The basophils secrete some chemicals. What is the chemicals? Heparin, serotonin. These all chemicals. Okay, histamines. Okay, these chemicals are produced by the basophils. Use eosinophils. This eosinophils, okay, increases at the time of the any inflammation, any allergic reactions at the time of the any dust particle enter into the nose. It causes to the allergies. It causes to the allergies. At the time of the allergy, which cells number is more? It is the eosinophils. At the time of the allergy. At the time of the allergy. Okay, eosinophils are increases, and also IgE immunoglobulins are increases. IgE immunoglobulins the number also increases at the time of the eosinophils infection. Any inflammation, any allergic reactions, which cells number is increases? Eosinophils. Okay, heparin, anticoagulant. Okay, and also histamines. Histamines. These are serotonins produced by the basophils. It is neutrophils, primary phagocytic cells, lymphocytes. Is act as a immunity. They will produce antibodies. The antibodies fight against the microorganisms. It's monocytes. It is also efficient phagocyte. They engulf the foreign material. Engulf the foreign material. Okay. It is the indication of the white blood cells. It is indication of the white blood cells. Thank you very much.